First of all, I would like to thank Professor Wagmare and Professor Partha for providing me the opportunity to present the work here. Today, I will discuss about the electrochemical sensors and how it works. So, before starting into details, I would like to describe about what is the overview of our lab and what are our expertise. So, first is the biomolecules modification. So, whenever we design any sensor, we use the molecules and modify it as per the requirement. Other things which we do in the lab is we develop the nucleic acid based assays using aptamers or the DNA, RNA, and other molecules which we develop by using the biomolecules modification is the antibody generation and development of immunoassays for that. The last thing is that the fabrication of devices for diagnosis, that I am going to discuss, that how we develop the devices for sensing different biomolecules. The next expertise is the targeting and drug delivery for diagnosis of certain diseases. So, what is the principle of sensors? As you can see from this the picture, that the transducer is the heart of or the core of the instrument or the device. So these transducers can be from the optical QCM or electrochemicals or anything that we would like to design. This transducer is connected to the biomolecules interface. These biomolecule interface could be anything like DNA, RNA, antigens, antibodies, or enzymes, or microorganisms if we are looking to detect the whole cell. And when it interacts with the analyte, which could be your antigens, chemicals, pollutants, DNA, or any food sample or vegetable samples, then it interacts with the receptor, and then the signal reaches to the transducer surface and it amplified through the amplifier, and the signal process in the form of the spectra or in the form of the readings that we would like to detect. So in this process, we start with the biomolecule modifications. Suppose this is one of the example. You can see this is one of the biomolecule, and then through the chemical synthesis processes, we generated the desired functional group at one end, and without disturbing the whole chemical structure of the molecule, and we conjugate it with the carrier protein, and this is how we link it. And after that, we characterize it with the SDL, like electrophoresis. We can see that as we increase the number of these molecules, there is a shift in the gel. And we also characterize this with other methods like fluorescence spectroscopy. You can see that as we are increasing the number of molecules, the fluorescence is going down. And later we validate it through the mass spectroscopy exactly to know how many number of molecules are there, and the suitable biomolecule then we use for immunizations in the animal model, and we collect the blood and purify the antibodies from the serum. These antibodies act as the biomolecules or the interface for development of any kind of assays or the sensors. So we check or evaluate the binding of these antibodies with the specific as well as the non-specific reagents, and calculate the inhibitory concentration or the limit of detection for certain molecules. Uh, Rather than doing the ELISA, which involves the HRP, which is enzyme based, we conjugate these antibodies with the further fluorescent molecules, which can enhance the sensitivity of the assays by tenfold. Then the second thing important which comes in the device fabrication is the nanomaterials. So selection of nanomaterial for the particular device is very important. So we synthesize the nanomaterial as per the requirement of the device, from gold nanoparticles to gold nanorods, quantum dots, silica, carbon nanotubes, fluorine, graphene, nanofibers, nanoflowers, silver nanoparticles, or iron oxide nanoparticles. So this is all depend upon the type of immunoassays or the device or the LFA that we would like to develop. So With this, what we have done, we developed a PDMS based device which is very flexible and easy to synthesize. So in this, we created the source and drain electrodes and put the gate voltage and calculated the change in the current over the period of time. In this, we use the three strategies rather. First strategy was that we used the carbon nanotubes because it is highly electroactive species, and then we immobilized the antibodies on its surface and flowed the antigen in the real-time manner. But in this process, we were not getting the enough change in the current, so then we reversed the strategy, put the antigen on the carbon nanotubes and then flowed the antibodies on it. So this strategy worked very well. So we went ahead with this strategy. Further, what we did, further to enhance the sensitivity, we labeled the antibodies with gold. So the combination of the carbon nanotubes together with the gold nanoparticles enhanced the sensitivity of the assay. So before doing all these analytical performance on the device, we have done all the characterization by AFM before and after conjugation of the antibodies with the carbon nanotubes. So this is the carbon nanotubes and this is the antibodies immobilized on the PDMS device together with the carbon nanotubes. And we have characterized with the fluorescence and CD spectra to see any changes in the fluorescence intensity if the binding is happening. Then we have done some in silico studies and to see whether the current is coming through the specific interaction or through the non-specific. So we were getting the specific interaction response. So this is how we made the device. There was a channel was created here, and then the CNTs were transfer printed onto this the lower PDMS device, and the channel was created. This is how, and the real-time measurements were done. So this is the scheme which I have explained a few seconds before, and here we were able to detect up to 100 femtograms per ml after using the scheme with gold nanoparticles and the carbon nanotubes. So with this, what we have done later on, we use the IgG antibodies by immunizing in the rabbit, and in this process, we have to collect the blood from the animal. So in this case, to avoid the collection of blood, to giving the not to give the stress to the animal or a non-invasive procedure, we developed the IgY antibodies in the chicken, and we have collected the antibodies from the egg yolk. Why? Because the IgY antibodies are reported to be more stable than the IgG. It is more stable because of the presence of extra CH2 domain, and it is more stable at higher temperature up to 60 degrees we have tested. And it is very easy to isolate IgY from the egg yolk, and you get the more concentration as compared to the blood from the one egg yolk. So with this, we developed the lateral flow device for this, and this is very simple. You put your sample into this sample well, and when it runs through the capillary action, it gives you development in the color on the strip, which is your antigen here. So with this, so what we have done, so these antibodies which we are seeing here are labeled one. So we have done taken the UV spectra before and after labeling of the antibodies with gold nanoparticles and characterize it through the TEM and develop the LFA for all four drugs, where we find that these antibodies, since polyclonal, are recognizing all closely related drugs with same sensitivity specificity of around 10 nanograms per ml. So to further process in this direction, what we have done, we have used the phage display library. So these phages are the bacteriophages that express the protein on its surface. So we have amplified the phages and have done the screening and sequencing of the bound phages. So these are the panning grounds that we have done through the library, around 48 or 50 panning grounds from each library, and then we have selected the highly bound phages from these libraries and checked their purity and sequence them. Then after that, we have labeled these scFv phages, which contains this VH and VL domain, and these VH and VL protein were conjugated with the particles and characterized through the DLS, UV, and TEM, where you can see the deposition of the protein surrounding the particles indicates that there is some proteins present, and hence the shift in the DLS hydrodynamic diameter as well as in the UV spectra indicated that yes, there is some binding happen. And after that we developed the LFA, where you can see that the sensitivity, the specificity of this drug was specific for two most closely related drug rather than the heroin and the codeine. So it is not binding with it. So while the specificity, sensitivity of this is quite similar to the IgY based assays. So then what the second FET device, which is the field effect transistors, what we have done in this, we developed this for the HIV's cardiac biomarkers and rheumatoid arthritis. So we have used the silicon dioxide substrate and put graphene on it, and created the source and drain electrode, and antibodies were immobilized on it, and the antigen were added, and real-time measurements were done, and change in the resistance were monitored. So during this process, the device characterizations were done to check the before and after treatment if there is any changes happen, and if the response is coming directly from the graphene or from the gold surface. So when we were using the gold, there was no response we were finding while adding the antibodies. So which means that the current which we were getting were very specific, and we have tested the stability of the device by keeping it for 4 weeks, and the device was quite stable at room temperature. And then we have done some characterization. This is the graphene optical microscope picture, and this is the SAM of the and this is antibody bound with the graphene. Then we have done the sensing, while in case of HIV it was 10 femtograms per ml, and CTN1 antibodies gave us the detection limit of around 100 femtograms, and RA antibodies gave us the detection limit of 10 femtograms per ml. Uh, then we have done the similar experiments for the Japanese encephalitis virus, which is the major problem occurred by the Culex mosquito, and in this case it affects pigs and kids in the northeast and the Bihar in India. So what we have done here, we developed the ELISA based assay and find out the what is the binding range and limit of detection in this case, and we have used GNS1 antigen in this study, and the limit of detection by ELISA was around 0.25 micrograms per ml, and we have fabricated the similar device, FET device, and characterized it in the similar way. And we have taken the other characterization like UVs, Raman, FTIR, and SEM for that. And the limit of detection through the real-time measurement was, in this case, around 10 femtomolar. So this is very, very sensitive and requires very less amount of sample, around 2 microliters, and we get the response within few seconds after adding the samples. So the next biomolecule which we use is the urokinase plasminogen activator receptor. This receptor overexpressed in the cancer patients. So we have done the sensing, used the antibodies specific for the UPAR, and flowed the UPA antigen. This UPA antigen has the specificity to bind with the UPAR. Because as per the literature, the UPAR binds with the UPA and vitronactin ligands and starts the its signaling mechanism. So with this, we have done the characterization after labeling of the antibodies with the graphene nanosheets, and Then the potential stat based measurements were done, where we have recorded the CV and DPV measurements, and at all the stages, like without anything on the electrodes, and after immobilization of the graphene nanosheets. And you can see in the red is the antibodies, and after adding the antigen, because of the specific interaction in the green peak, it goes up. Then before testing the analytical performance of the device, what we have done, we have done all the optimization, like different concentration of the antibodies, response time, temperature, pH, and the scan rate, and we were able to detect up to 10 femtomolar of U per antigen in this case. And apart from this, we have done the testing or electrochemical detection of certain pesticides by using the cantilever based, which measures the change in the deflection due to change in the mass on the surface, where we have used the gold coated cantilever and functionalized the gold with the SATA, because gold is inert and cannot bind with any moieties. So after that, we have flowed the atrazine and were able to detect up to 1 ppt of atrazine. Then we have tested the FET based sensor for the chlorpyrifos and 2,4-D, and another electrochemical device for chlorpyrifos. Then this is the very interesting study we have done for the aflatoxin M1 detection, where we have fabricated the paper microfluidic device that anyone can take to the field, and can just put the sample, milk sample here, and by seeing the change in the color can tell if there is a aflatoxin or mycotoxin is present in the sample or not. So what happened in this case, the principle is that we use the particles and use the specific aptamers in the, then these aptamers binds through physiosorption on the surface of gold, and after adding NaCl or the salt, it has the property that because aptamers are bound onto the surface, then you will not see any change in the color, and the color of the solution will be pink. Once you add the specific molecule to this aptamer, these displacement occurs, these aptamers will go and bind to the specific molecule, and due to the presence of salt, the color will change into blue. So this concept we applied on the paper microfluidic, and this is how we detect it. So this is another biomolecule which is very, very interesting. We developed is by using the peptide based sensor, which we call as activatable nanosensors, because we know in the cancerous patients there is overexpression of proteases. So there are certain proteases for certain types of cancer. So these protease recognition site we put in the middle of this peptide, and the N and C terminal of the peptide were labeled with the biotin. So then we made the iron oxide nanoparticles, which were labeled with these streptavidin. So due to the interaction between biotin and the streptavidin, there was aggregation of the molecules that one can see through the naked eye also, and as well as through the MPS, which is the magnetic particle spectrometer. So the signal goes down when there is aggregation, and when we add the cells which is expressing the proteases or the cancer cells, there is a cleavage occurs in between these this peptide, and there is a disperse particles get dispersed, so the signal goes up. So this also can be seen through the naked eye. This was one of the study. So and then this is the iron oxide nanoparticles which we have used for the targeting, and we have seen that these specific particles, when labeled with these specific receptors like lactoferrin, are able to cross the blood-brain barrier and can see the targeting by confocal as well as through the Prussian blue stains. So this is the MPS-based signal that we concluded that after 12 hours, at 12 hours, we can see the response of these particles. So this is the very recent study we have done. So here we have used the urokinase plasminogen activator receptor. This receptor is having a GPI linker that does not cross the cell membrane, and this receptor binds with the UPA and vitronactin. So these two ligands we synthesized and labeled the gold nanoparticles with the polymer, and functionalize it with the peptide. This peptide is conjugated with the FITC. So we can see the what is happening inside the cell in terms of targeting. So in this case, we have used three different combinations. First, we have used the UPA ligand, which is GFD. Then in the second experiment, we have used the SMB ligand of the vitronactin. And in the third experiment, we have used the combination of two ligands, GFD and SMB. I'll not go into these results, but with this imaging, you can see there is a efficient targeting happened in case of both ligands together, GFD and SMB, as well as with the facts, we can see these results. So this is the very recent studies. And in the end, I would like to thank uh, Indian Academy of Sciences. Again, because of that, I received the SCRB Women Excellence Award this year. I really thank Professor Wagmere and Professor Partha for providing me the opportunity. Thank you so much.
Sonu, there is a question from YouTube by Professor Bhaskaran. One hears about handheld optical biosensors for COVID test on patients. Is that something you can comment on?
Yes, sir. Actually, these optical biosensors, we can use that, but the sensitivity in this case will be little lower. So in this case, if we would like to develop the handheld device, the electrochemical sensors will be better.
I had one curious question. Sonu, how do you make sure that different molecules may not give you very similar response in terms of change in resistance?
Sir, different molecules, if it is not having the similar structure, then it will not bind to the antibodies, because antibodies have the specific epitope that it binds to the specific antigen only. So so if the antigen or that biomolecule will not bind to the antibody, we do not get a response. We get a straight line.
I
So
see.
if there is a specific molecule, we get a dip in the resistance
I see.
immediately. Yeah.
Thank you very much, Sonu.